This part is cyclohexyl 1,2-diamine. So we have two molecules of cyclohexyl 1,2-diamine. In one part we have connected them with the ethylene moiety, and in another part we have connected these amines with 2,6-dipicolyl moiety. So we have a complicated 5 coordinated or 5 pentadentate ligand, and manganese again here in the plus 2 state. And it forms a 7 coordinate complex whereby the 5 nitrogen atoms of the ligand is coordinated in a plane, and below the plane and above the plane there are two chloride ions coordinated to manganese. So this complex is found to be most reactive in terms of superoxide dismutase mimicking activity, and probably it will be commercialized in near future. Any question here? Okay. Next, we shall go to application of vanadium complexes as insulin mimetic agent. That means those who have got deficiency of insulin, for them variety of treatment up there. Mainly they get injected with insulin, but nowadays gradually some compounds are also coming into market. These are in the very advanced stage of research or has been commercialized to a limited extent. Some vanadium complexes which can mimic the insulin property. That means the glucose hydrolysis property, right? And the most promising one is this complex. Here vanadium is in the +4 state, and the ligand, this is called maltolato ligand. Maltol, coming from maltose, right, or malt. So maltol is a standard, is actually is a natural product, and it is used in food additive, right? It adds a good smell to the food sometimes. In some food there is some maltol added as a food additive, and malt is actually consumable, right? So human consumable. So from malt, this particular compound is derived. It is called maltol, and when it is deprotonated, it is called maltolato. So this part, why we are looking for natural products? Because then, if we can complex metal ion with a natural product, there is a chance that it will be more biotolerable than if you take a synthetic ligand. So this is a natural ligand with which two such ligands are coordinated to vanadium. And so vanadium, you see, this is 1 minus charge, this is 1 minus charge, and this is 2 minus charge. Vanadium is in the +4 oxygen state. So you have a vanadium 4 bis maltolato oxido vanadium 4 complex, right? So these are maltolato ligand, and we have the oxide ligand. So bis maltolato oxido vanadium 4 complex, and it is it has got very good insulin mimetic property. This is more suitable because it can be administered orally. It has been found this drug can be administered orally. Apart from this vanadium maltolato complex, there are other vanadium complexes which are also available commercially and or in a very advanced stage of research. Again, we are looking at vanadium complexes with natural ligands, right? Ligands which are available in nature. And here we have it is nothing but this ligand is nothing but a derivative of natural amino acid called cysteine. So cysteine is one of the, there are two sulfur containing amino acids in our body, cysteine and methionine, and this is a derivative of cysteine. When it is cysteine, we have OH here, and here we have a derivatized, so that the lipid solubility increases. It can go inside the cell easily, and in order to increase the lipid solubility, here what have been done, that OH is methylated, so it is methylated cysteine methyl ester, right? So a methyl ester has been formed. So vanadium bis-cysteine methyl ester complex, where again dioxo group is there and one amino group, and one sulfur from the each of the two amino acids are coordinated to vanadium. So this is also a very good insulin mimetic agent, and very close to that compound is this one. Again, this is cysteine. You see cysteine. The only difference is that here, instead of this carboxylic acid in cysteine, this has been converted to a organic amide with 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 8 carbon atoms. The objective, as I have told you in one of my last class also, when we are discussing the plutonium chelating agents, Puchel, that the objective of attaching a long chain hydrocarbon to this compound, to this original polar compound, is that it becomes more covalent, right, and it's become more lipophilic, so that it can penetrate the cell wall and go inside the cell. So very similar compound. Here it is a very simple model where we have the methyl ester, and here we have a amide of cysteine, and it has been this amide is converted using a long chain hydrocarbon, so that the lipid solubility increases. And again, the commercial name of this compound is Naglivan. So again, it is actually commercially available, this compound. And then finally, we have another vanadium compound, which is here this group in, then two alkyl groups, and C double bond S minus. This is called dithiocarbamate, right? This group C double bond S minus N, and then two alkyl groups or two aryl groups. These are called dithiocarbamate. So here we have two dithiocarbamate ligands coordinated to vanadium 4. Vanadium is again, there is oxido ligand, two sulfur ligands, and this part is called pyrrolidine, right? But from the pyrrole, you reduce pyrrole to pyrrolidine. So pyrrolidine dithiocarbamate, bis ligand of bis complex of this ligand is used as insulin mimetic agent. So you will be asked to name two vanadium compounds which can show insulin mimetic properties. So among these four complexes, which any two which you like, but I will advise to keep BMOV, this mannitol, and you can take example from any three of this, any one from these three. But I shall again advise this one to remember, because this has been commercialized. Then we go to certain gold 1 compounds, which have been used as medicines for rheumatoid arthritis, right? For arthritis. And again, our ligands are naturally derived. Now, if instead of S here there is OH, this was glucose. So this is glucose, except that here one of the OH groups has been replaced by SH. So you can say this is a thioglucose, right? Monothioglucose. One of the OH group of the glucose is replaced by SH. And why SH? Because I have told you that soft hard acid base theory, or hard soft acid base theory discussed in last class. Gold, particularly gold in the plus one oxidation state, is highly soft metal ion. And so if you want to complex gold, you require relatively softer ligands. And I have told you, compared to oxygen, sulfur having a larger size and diffused electronic cloud, sulfur is a much softer ligand than oxygen. And that's why, in order to get good strong coordination with gold, one of the OH groups has been replaced by a thiol group, and now it can coordinate to gold in this fashion, right? So we have a gold 1 thioglucose complex whose commercial name is solganol, right? So commercially it is known as solganol. There is another derivative of this complex which is again marketed by two different medicinal companies, one under the brand name of Oranofin and another under the brand name of Reduera. What is the difference between these two? You see, They are identical complex, except now the OH groups has been acetylated. So it has been converted to the acetyl group. OH, instead of OH, we have OAC. And gold, uh, whereas it is coordinate to sulfur, it is also coordinate to a triethyl phosphine ligand. The whole purpose of this is that compared to this compound is more lipophilic. It has got more covalent like character, right? The more there is less polar compound this is. As a result of this, this compound is more lipophilic, and because it is more lipophilic, so it can, I am repeatedly telling you, lipophilicity is urgent requirement so that it can penetrate the cell membrane and go inside the cell. So the cell penetration power, right, permeability through the cell membrane to go inside the cell will be greater for oranophine or reduera compared to solganol. And we have another gold compound. If you have a OH group here, it was malic acid. So instead of malic acid, we have a thiol group here. So it is thiomalate, and we have a gold thiomalate complex, which can be also used for actually anti-rheumatoid arthritic treatment. But the most important compounds are these two, solganol and oranophine or reduera. Now, though the how these compounds really act, or what is the mode of function of these gold complexes are still under a lot of research. But from whatever the modern research has has actually revealed, we can say that these gold compounds go to the lysosomes in our body, and in the lysosome these gold compounds prevent release of pro-inflammatory cytokines. Now in COVID, we were always seeing in the TV or discussions that cytokine storm actually kills the patient. So excess of the immunosystem reaction, that actually sometimes is quite bad. So here there are certain pro-inflammatory cytokines which actually are responsible for inflammation and other other kinds of diseases. So it controls the release of pro-inflammatory cytokines and thereby reduces pain. And so these gold compounds are very good for as anti-rheumatoid. In fact, there are only few compounds which are available for as rheumatoid arthritis drug. So gold complexes is very important. Gold one complexes are very important in that aspect. Next, we shall pass on to the probably the most important application of metal complexes in medicine. That is metal complexes in cancer chemotherapy, right? So maybe I shall just give an introduction today, and then we shall discuss it later, because today I have a meeting, so I will leave somewhat early, 10 to 15, 10 minutes early. So, first of all, what is cancer, right? And now this thing I have actually taken from the National Institute of Health website, with the NIH USA website. So in NIH USA, it describes cancer as a disease which arises from the uncontrolled cell division in some parts of the body. Normally in our body, the cell division and cell death, they are very strictly controlled, right? They are very tightly controlled. But due to some disorder in your body, which often actually arises due to some genetic modification, either from one generation to other generation, or due to exposure of a person to certain kind of chemicals or radiation, what may happen, their DNA, their genes may get modified. And due to the modification of the gene, what will happen, this orderly process of cell division and cell death, it breaks down, and as a result of this, old cells do not die and new cells are produced in an uncontrolled fashion. And whenever this kind of things happen, that means the control over the cell death and control over the growth of the new cells are lost, then it may lead to formation of tumors which may grow cancerous. Very often what happens that cancer cells can invade and spread to nearby tissues, right? Unlike benign tumors are localized in one particular space, whereas malignant tumors, they may spread to different parts or different nearby tissues, so thereby increasing the infection or increasing the possibility of being fatal. Right. Sometimes cancer cells can also break off from original tumor site and travel to some distant places through blood or lymph system and produce tumors there, which are called metastases. So in fact, it has been found that if you try to catalog how many patients are dying due to primary cancer and how many patients are dying due to metastasis, very often you will find that there are more number of patients who are dying due to metastasis than due to original primary cancer. So metastasis is a very dangerous thing whereby some cancer cells break off from the original site and they are transmitted through the blood or lymph system, and then they get stuck up in certain other parts of the body and grow tumor cells there, cancer cells there. So you require anti-cancer agents which attack the primary cancer, and you require anti-cancer agent which can check the metastasis growth. Cancer, according to NIUS site, is a genetic disease. This genetic disease may be inherited. That means this defect of gene which causes cancer, which causes the breakdown of the discipline regarding the cell growth and cell death, that may be inherited or it may be induced by external or environmental stimuli such as exposure to carcinogenic chemicals or certain radiations like UV and X-ray radiation, etc. So in any case, cancer may be inherited, cancer may be stimulated by exposure to carcinogenic chemicals or certain hazardous radiation, and initially the some defect in gene occurs, and this defect in genetic system then leads to uncontrolled cell growth and cancer. In general, cancer cells have more genetic changes and such than normal cells, right? So cancer cells has got cancer. If you compare the normal cell and cancer cell of a person infected with cancer, you will find that the cancer cells contains more genetically modified cells than the normal cells. Now, here we have described, all of you know that the DNA has got a double helical structure, and this DNA, this is called one strand, this is called other strand. So there are two strands which are interlocked by hydrogen bonding, and you know probably that A, T, G, C, etc. So there are certain amino acids, certain bases, right? Adenine, guanine, cysteine, thymine, etc., cytosine, thymine, etc., which actually uh, are responsible for this kind of complementary hydrogen bonding, and this hydrogen bonding holds the two strands of the DNA together. Now, what is the objective of the metallocancer drugs? Metallocancer drugs uh, usually target the DNA. They target the DNA and they modify the DNA in such a way that they distort the DNA, they elongate the DNA, they unfold the DNA, the double helix unfolded in such a way that DNA replication is stopped to a great extent. And when DNA replication is stopped, that means cell multiplication is also stopped. So this is the approach taken by the most of the metallomedicines, that you develop certain systems which can target the DNA, and in targeting the DNA, it should modify the DNA in such a way that the characteristic of the DNA will be changed, so it will undergo more elongation. The number of turns will decrease, right? It may be unfolded, etc. So lots of changes, physiochemical changes of DNA will occur. And as a result of this kind of DNA, distorted DNA, cannot participate in DNA multiplication. And if DNA multiplication is stopped or inhibited, then cell multiplication will also be stopped. Now, in the second slide, what I have shown is what are the different ways by which a metallomedicine or a metal complex can bind to DNA, right? Now, here, for better understanding, we have marked the two DNA helix with two different colors. So one helix is marked with green color. Sometimes this helix, one helix is also called strand, strand, strand. So it is a double-stranded DNA. One strand is marked with, this helical strand is marked with green color, and the other helical strand is amber color. And then we have we have shown the different modes of attachment of metal complex to the DNA. Now, in the extreme left, in the upper side, what do we have? That a metal complex attacks DNA in a monodentate manner. That means only one strand, only one base of the DNA helix, right, is coordinate to the metal ion. So it is a monodentate coordination to only one strand. So this is relatively uncommon, right? So, but in there may be certain cases where only one strand of DNA is attached to the metal complex in a monodentate fashion. Usually, this kind of binding does not lead to high anti-cancer activity. But this kind of binding you may find in some cases. Then, if you go here you find that metal complex has bound DNA in a bidentate fashion, right? So, two coordinate sites of metal are bound to DNA. Now, this binding, first of all, you see, it has occurred, this binding has occurred to the same strand, right? So, both the sides, both the coordinate sides of the metal ion are coming from this green strand, right? So, it has attached itself to the base of the green strand. So, it is called intra-strand binding. And since it is this binding, two binding sides are next to each other, it is called 1,2 intrastrand binding. That means it is binding to two successive bases of the strand, and so it is 1,2 intrastrand binding, right? On the other hand, here there is some similarity, there is some dissimilarity. Similarity is that here also metal complex, two coordinate site of the metal complex is obtained by two bases from the DNA double helix. But here one coordination is coming from the amber color and another coordination is coming from the green color. So here it is binding binding the double helix in a inter-strand binding, right? So it is inter-strand binding. So it is connecting one strand, this amber strand, with the green strand. So it is called INTR, inter-strand binding, right? So so you have three modes. Then the other three modes may be that metal complex, one coordinate side of the metal complex is occupied from one base of the DNA, and the other coordinate site may be occupied by some other donor atom from a protein, nearby protein. So one is bonded to the DNA strand and other is bonded to a nearby protein, right? So it is some kind of protein DNA cross-linking, right? So metal complex is cross-linking a protein with a DNA. This is again relatively rare, right? Here we have metal complex binds to two strands, but these two strands are coming from two different DNA double helix. So here again, metal complex is binding through two of its coordination sites, but these two coordination sites do not belong to the same DNA double helix. One coordination site is occupied by this double helix, and another coordination site is occupied by another double helix. So it is a it is a cross-linking between two DNA double helix. Here it is cross-linking between one double helix and one protein. Here it is cross-linking between two DNA double helix. And finally, here again it is a intra-strand binding because it is binding to the green strand. But instead of 1, 2, here you have 1, 3. The class. It was that one. So it is 1, 2 or one. It is one instead of 1, 2 here. Instead of 1, 2 here, it is 1, 3. Okay. So 1, 3, 1, 3 binding. So these are the different modes of binding of the DNA double helix by a metal complex. And I shall end today's discussion with only one compound. This compound, this is the classical compound called cisplatin, which is cis-dichlorodiamine platinum. This is the very first generation anti-cancer medicine, which was discovered by Rosenberg in around late 1960s. Now, what Rosenberg was trying that he was trying to find out the how electric field can affect the cell growth. And as a result of this, Rosenberg has taken some bacterial cell called E. coli cell, Escherichia coli cell, in a bacterial medium, where we have in a medium where we have as a culture broth, we have certain ammonium chloride, water, and then he applied small electric field using two platinum electrodes. Now, Rosenberg, after applying electric field for overnight, he found that the cells are dead. Right, so he found that there is a filamentous growth of the cells. That means cells are elongated. It has become filamented, right, fibrous, and the cells are dead. No more cells are alive. So Rosenberg tried to analyze whether it is due to the electric field or some other effect. And so what he did, he changed the electrodes with other metal ions or graphite, etc. He found that for when electrodes are other metal ions, like say copper, right, like say nickel, or say graphite, it is found that there is almost no effect under identical condition. There is no effect on the on the bacterial cell, but only when platinum electrode is used, the cells die. Then Rosenberg actually postulated that there must be platinum must be reacting somewhere with the metal with the bacterial cell, and he analyzed the growth medium, and from the growth medium he isolated this complex PTNase32Cl2, PTNase32Cl2, yes, which is called cisplatin, and as a result of this, this complex was discovered, cis-dichlorodiamine platinum. It was discovered. Okay. Now this is still quite a effective compound for treatment of cancer, but it has got many side effects. Particularly, it has got high nephrotoxicity and neurotoxicity. Nephrotoxicity means kidney toxicity, and neurotoxicity you can understand. So, and it has also got low solubility, etc. So, people have searched for newer anti-cancer compounds, and they have stuck upon many other platinum compounds and other metal complexes. But I shall stop it here because I have a meeting. People are calling, so I shall be stopping here. Thank you.
Sir, can you please share us the PPTs? It will be helpful to study.
No, what I have said you that because the recordings are not available, so what I shall do after I finish this particular part, I shall take recorded lecture of earlier years. Okay, earlier years, and then I shall actually send you those recorded lectures. Okay.
Okay, sir.
Okay, sir.
Thank you very much. So I am leaving.
Thank you, sir.
Thank you, sir.
Thank you, sir.